Welcome back to Advent and a very interesting episode on Dr. Mohanty's Masterclass. After discussing nitrosamines in my previous talk, today we move to an important related class of impurities, that is nitrosamine drug substance related impurities or NDSRIs. What are NDSRIs? Unlike common nitrosamines, which are often formed as byproducts during manufacturing or storage, NDSRIs are structurally linked to the drug molecule itself. They are generated when a nitrosetting agent reacts with specific functional groups present in the active pharmaceutical ingredient API or its intermediates. Because of this close structural relationship, NDSRIs are unique to each drug substance. This makes them more complex to identify, predict, and control compared to simpler nitrosamines. Regulatory agencies such as US FDA and EMA have recognized their potential carcinogenic risks and have issued strict guidelines for risk assessment, control strategies, and development of reference standards. In essence, NDSRIs represent the next frontier in nitrosamine control, requiring a deeper understanding of chemistry, drug structure, and analytical methods to safeguard patient safety. What are the sources of NDSRIs? NDSRI can form during the manufacturing process of pharmaceuticals through various pathways, which includes reaction of secondary or tertiary amines with nitrosetting agents. For example, secondary or tertiary amines present in drug substances or excipients can react with nitrosetting agents such as nitrites, leading to the formation of NDSRIs. Some drug substances may degrade over time, leading to formation of NDSRIs. Third, raw materials contaminated with NDSRIs or nitrosetting agents can introduce these impurities into the final product. Fourth one is cross-contamination. Cross-contamination during manufacturing process can also lead to the presence of NDSRIs in drug products. So what are the regulatory guidelines for NDSRIs? Regulatory agencies such as US FDA, EMA, and others have issued guidelines to control and limit the presence of NDSRIs in drug products. The key points include risk assessment, where manufacturers must conduct a thorough risk assessment to identify potential sources of NDSRIs. Analytical testing, like validated analytical methods, must be used to detect and quantify NDSRIs in drug products. Control strategies must be implemented to mitigate the formation of NDSRIs during manufacturing. Fourth one is acceptable limits. Adherence to the specified acceptable intake limits or AI limits is essential to ensure patient safety. However, in the case of NDSRIs, this poses a significant challenge since specific toxicological data is often unavailable. As a result, establishing scientifically sound AI limits becomes difficult and remains an evolving area of regulatory and scientific focus. So, what are the control and mitigation strategies for NDSRIs? To control and mitigate the formation of NDSRIs, manufacturers can implement several strategies. Say process optimization. So, manufacturing processes have to be optimized to minimize the use of nitrosetting agents and amines. This remains a challenge as many APIs contain secondary and tertiary amine functionality. Second is raw material quality control. To ensure that raw materials are nitrite free and tightly qualified. Third is environmental controls, which must be implemented to prevent cross contamination. Also, to conduct stability studies to understand the degradation pathways and potential for NDSRI formation. Last but not the least is adjusting formulations to replace or reduce the use of amines or nitrosetting agents. Besides, storage and packaging conditions must prevent degradation pathways. Analytical monitoring should be ongoing throughout the product life cycle. Most importantly, proactive risk assessments and supplier qualification programs help mitigate issues before they reach patients. The most important point we come to is detection and analysis of NDSRIs. NDSRIs, as I told, are structurally similar to parent drug, making their detection and separation more challenging than common nitrosamines. However, following advanced analytical techniques are used for their detection and quantification. Most important is LCMS, that is liquid chromatography tandem mass spectrometry. This is the most widely used technique for identification and quantification of NDSRIs. Second is high resolution mass spectrometry, which helps in structural elucidation and confirmation. Third is GCMS, that is gas chromatography mass spectrometry, which is useful in specific cases, though less common due to the non-volatile nature of many NDSRIs. When it comes to method development for NDSRIs, the challenges are quite unique. First, because they are structurally related to parent drug, separating them from main API can be very difficult. We need highly selective and sensitive methods, often capable of detecting impurities at parts per billion ppb levels. Second, there is the problem of false positives. The drug matrix can interfere with the detection, so methods must be designed to avoid misidentification. Finally, robust sample preparation is critical. In some cases, we may need enrichment or selective extraction to reliably detect these impurities, and what we are doing in Advent for NDSRIs. Another key requirement in analyzing NDSRIs is the availability of authentic reference standards. Without them, it becomes extremely difficult to validate analytical methods or to accurately quantify impurities. Since NDSRIs are unique to each drug substance, ready-made standards are not always available. That is why pharma companies often depend on custom synthesis or specialized suppliers like us, Advent, to provide these materials. Once in place, these reference standards give confidence, both scientifically and from a regulatory perspective, that the analysis is accurate, reproducible, and compliant with global expectations. At Advent, we have taken a pioneering step by developing a wide range of NDSRIs as working standards. These standards are being increasingly demanded by pharmaceutical companies worldwide, as they are essential for quantifying nitrosamine impurities and validating their drug products in line with regulatory requirements. By supplying these reference materials, we enable our partners to meet compliance expectations, accelerate product development, and ensure patient safety. This initiative positions Advent as a trusted global partner in nitrosamine impurity management. Here are some of the representative examples of NDSRIs, of APIs, and related substances or intermediates with structures. Most of them are in ready stock, available with high purity. Customized packs are also available from milligram to gram scale. Certificate of analysis is provided along with all characterization data such as mass, IR, proton NMR, carbon 13 NMR, etc. Please get in touch with our sales colleagues or channel partners for your requirements. To conclude, NDSRIs present a significant challenge for the pharmaceutical industry with implications for drug safety and regulatory compliance. By understanding the sources of NDSRIs, adhering to regulatory guidelines, and employing robust analytical and control strategies, manufacturers can effectively manage these impurities and ensure the safety and efficacy of their drug products. Continuous vigilance and advancements in analytical technology will be crucial in addressing the evolving landscape of nitrosamine impurities. At Advent, we remain committed to supporting our partners in navigating this challenge with a mission to enable safe, compliant, and future-ready medicines. Thanks for watching this special episode on NDSRIs. Stay tuned, stay curious, and explore the wonderful worlds of chemistry. Thank you very much.